Antiviral drug remdesivir has been cleared for treatment of COVID-19 patients under emergency use. And this was stated by Union Health Ministry yesterday. So, in this context, it becomes important for us to understand the mechanism through which this drug acts, and especially against the coronavirus. Although this is not a new drug, and it was not discovered to target the COVID 19 virus, but more into that later on. But first, let us understand how remdesivir acts upon the virus. And for that, obviously, we need to understand how virus itself enters the body and replicates. But first and foremost, it needs to be cleared at outset that remdesivir is not a vaccine, and so it cannot prevent an infection. Instead, it works by attacking the virus once it is already spreading inside the body. So let us first understand how a virus interacts with the cells of our body. Now, the first stage obviously is the entrance of a virus into a cell. Because unless and until a virus enters a cell, it cannot replicate itself, because virus need the cell's protein making machinery to multiply. So they first need to gain entry into a healthy cell. And as you might know, that coronavirus or COVID-19 have a special set of proteins known as spiky proteins that allow them to bind with the cells. So as you can see in the animation, that the spike protein helps the virus to latch on to human cell. Then after it latches onto the cell, the virus fuses with the cell and once inside releases a strand of RNA. Now, RNA or ribonucleic acid is like a blueprint, which is basically a set of genetic code that has instruction to make exact copies of the same virus. So as you can see in the animation, the virus has already released the yellow or orangish color RNA into the cell. Now, the reason why the virus has entered into the cell is that it wants to create more of its copies. And in that process, it is helped by ribosomes, which are tiny particles in the host cell, which have this special capability to read genetic material. And as you can see in the animation, as the virus's RNA passes through the ribosome produces exact viral proteins which the virus wants this to produce. Now, these production of viral proteins is much needed because they are the essential ingredients of viral RNA as well as other parts of the viruses like the outer spike protein itself, which in the first place helped the virus to enter into the cell. The proteins are also needed to make the outer membrane of the virus as well. So as you can see in the animation, as soon as the viral RNA interact with the ribosome, not only it is producing the RNA copy, but it is also producing the viral proteins. So the component in orange color will form the genetic material of the virus, whereas the component in the red color which is being produced will form the spike protein. And this you can see will lead to the formation or assembly of a new virus inside the host cell. And thousands and millions of more such viruses are produced inside the healthy host cells. When fully assembled, the virus can now exit the cell to seek other healthy cells and start the process again. And this goes on and on unless and until the body develops immunity against the virus. Now the question arises, how does remdesivir helps in cases of COVID-19 infections? Well, it is not difficult to think that it stops this process of replication somewhere in between. And that is, as you can see in the animation, remdesivir mimics a part of viral RNA itself. And during the copying process, it inserts itself into RNA strand. So as you can see in the animation, the black color circles are remdesivir and they have comfortably placed themselves in the RNA copies which are being produced. And remember that these copies are going to form the RNA of the new virus which is being produced inside the host cell. When attached, the drug prevents any further copying, leaving the RNA strand incomplete and unable to produce critical virus parts. Now, as you can see, that the process of replication of virus has been hampered by the drug because the entire replication process was slowed down. This one black circle, which is the remdesivir molecule, has prevented the formation of a complete virus. And hence the entire replication process slows down because the remdesivir has placed itself in almost all the viral RNAs And defective viruses with partial RNA cannot replicate in other cells, because to replicate they need the whole genetic material. But as you can see, instead of all the circles being orange color, there is one circle with black color, and this has stopped the entire process. So this is how remdesivir works. So finally, we have compiled everything in a single page so that you can take a screenshot of it. Remdesivir, as we have stated, is not a new drug, but rather it was initially developed to treat hepatitis C and respiratory syncytial virus. Now, it was also one of the most probable drugs for the treatment of Ebola as well as the SARS and MERS, two other coronaviruses that infect humans much like the way COVID does. Although it never made it to the approval stage for those uses, people all across the world are desperate for options to mitigate the COVID 19 pandemic, and they are looking towards older drug so that it can be repurposed against COVID 19, and hence they started the trial for remdesivir. And it is great to know that in the instance of COVID 19, this miracle drug has reached the regulatory approval stage, becoming one of the most promising therapies against COVID 19 till date. And as you know, that as far as the actions are concerned, as soon as the virus enters the cells after its protein known as spike latches on to the cell's outer membrane, then virus releases the genetic code, which is then held by the ribosome for the replication process for the formation of proteins which form the genetic code as well as the other proteins. And utilizing these various proteins, a new virus is assembled. Now, what remdesivir does is that it interferes with the replication process over here by getting itself inserted into the RNA, so that the final copies of the viral RNA which are being made are rendered useless and defective, and hence the process of replication stops then and there. Let's now move on to the next news. Now, this next article appears on page number 15.